Okay, good afternoon. Till now, we have seen about different groups of insecticides, and from on today's class, we will discuss about bioassay. That means a way to determine the toxicity of the insecticides. What are the ways to determine the toxicity of insecticides? What are the uses of bioassay? And what is the basic principle of bioassay. Uh, I wrongly asked you, posted the probit analysis part earlier because unless and until we complete the bioassay part, then only we can go for probit analysis. I thought I have completed it. Okay. So in the next class, tomorrow we will take up probit analysis. Today we will be taking up insecticide bioassay and its applications. Okay. What is bioassay? The bioassay, as the name indicates, bio means life, assay means measuring. So, as measuring of determination. So, the basic definition of bioassay is that it's a procedure used to determine the relationship between a physiologically active substance. Physiologically active substance means an insecticide, any toxicant, even a drug. Say we are discussing about the COVID-19 vaccine. So we will be studying the relationship between the vaccine, that is a physiologically active substance, and the effect which they produce on insects or living organism. And if you are injecting the vaccine to a human, what is the effect? Whether the antibodies are developing, whether there is any adverse reactions. So all these things comes under bioassay. So, in a broad sense, bioassay it is the procedure to determine the relationship between physiologically active substance and the effect it produces on insects or any living organism. So, we'll be injecting or we'll be delivering a physiologically active substance, a drug or a pesticide or anything, and we will be measuring the effect of that physiologically active substance or an insecticide or a drug that will be produced in the living organism. So that is a broad definition of bioassay. Coming to insecticide bioassay, it is defined as the quantitative procedure. It is a quantitative procedure because we measure the response. Well, how many insects are dead, how many insects are alive, we count it. So it's a quantitative procedure, it is which is used to determine the relationship between the amount of insecticide. This can be dose or concentration. We'll discuss about the difference between dose and concentration later in the subsequent slides. So insecticide bioassay, it is a quantitative procedure which studies the relationship between the amount, the quantity of the pesticide, which can be dose or concentration, which is administered into an insect, and the magnitude of the response in a living organism. So we can define bioassay as the study of the relationship between an insecticide and the response in a living organism. In the case of insecticide, the response in an insect. So it is a quantitative procedure. That means we measure the numbers. It studies the relationship between the amount of insecticide administered, the amount, the dose, the concentration, the quantity of insecticide administered and administered to the insects, and we studied the relationship between the quantity of the pesticide and the response it produces in the insect, the magnitude of response. Magnitude of response means how much insecticide got killed, how much insecticide is alive. So the insecticide bioassay determines the relationship between the amount, that is dose or concentration of an insecticide administered to an insect, and the magnitude of the response the insecticide produces in an insect. So in a nutshell, we can say that bioassay is study of dose response relationship. That means we will administer a specific dose and we will study what is the response, what is the magnitude of the response in a living organism. So toxicology, bioassay is nothing but the study of dose response relationship. Because of this, toxicology is also called as science of doses. Which dose to be applied? So at which dose, what will be the response in the organism? So this is bioassay is used to test any toxicant. It can be, I told you, it can be insecticide, fungicide, herbicide, etc. Anywhere we use bioassay. Even for human drugs, we use bioassay, wherein we studied the relationship between the quantity of the drug and the response it produces in the human being. So that is bioassay. And for coming to the insecticide bioassay, here it helps to determine the LD50, that means lethal dose 50, LC50, that is lethal concentration 50, LT50 is lethal time 50. We will discuss about the difference between LD, LC, and LT in the next slide. Next one is KD50 or knockdown dose 50, knockdown concentration 50, knockdown time 50, effective concentration 50, effective dose 50, persistent toxicity, and relative toxicity. We'll discuss about all these things in the next upcoming slides. LD50. What is LD50? LD50 is the amount of poison per unit weight of the organism. That means we know the exact quantity of the pesticide entered into an organism, and we can specify the quantity as what is the how much quantity of pesticide, that means milligram of pesticide per unit weight of the organism. So the LD50 is always expressed as milligram per kg. Here one precondition is that we should know the exact quantity of pesticide that is delivered into the insect population, delivered into the test population. So we take up a test method by which we can precisely know how much quantity of pesticide is entering into the insect body, and we will measure the mortality. So LD50 is the quantity of the poison or an insecticide per unit weight of the body weight of the organism, which will kill 50% of the test population. That is nothing but LD50. The condition is that we know the exact quantity of pesticide per unit weight of the organism, which will kill 50% of the test population. So that 50% of the test population that you might be familiar. Coming to the next, that is LC50. LC50 is the most common one which we use. LC50 is we prepare different concentrations of pesticides, say 0.5 percentage, 0.01 percentage, 0.05 percentage, etc. And we apply the pesticide into the insecticide or to the insect or the test population, and we measure the mortality, and LC50 is the concentration of the pesticide which will kill 50% of the test population. But one difference between LD50 and LC50 is that in LC50 we cannot determine, we cannot say that we have sprayed a 0.05% solution of any organophosphorus pesticide into it on the body of an insect. We have sprayed the pesticide, but we cannot precisely say that what is the exact quantity of the pesticide that entered into the body and killed 50% of the test population. We can only say that yes, I have sprayed this much concentration of a pesticide and this dose killed 50% of the test population. Whereas in LD50 we can say that I have administered this much quantity of exactly this much quantity of the pesticide into the body of the organism, so that the dose is how much this quantity of pesticide per unit weight of the insect, and it killed 50% of the test population. So LC50 is usually expressed in ppm or percentage. Here, the pesticide will be dissolved in an external medium. That means pesticide usually we dissolve in water and all. So that is the term external medium over here. Then coming to next, that is LT50. Your senior, the Deepthi Madam student Jansi was working on endomopathogenic fungi, and you might have said, what is the time required for the mortality of an endomopathogenic fungi? How much time an endomopathogenic fungi takes to kill an insect? Roughly, no. Why? No answers. 2 to 3 days. No. Fungi, endomopathogenic fungi. 10 days, Rahul. Okay. If the humidity is low, it is 10 days. Okay. Roughly, it is 7 to 10 days. We usually say it is 7 to 10 days. You remember it. So, so if you are trying to compare different strains or isolates of an entomopathogenic fungi, Jansi had different strains of entomopathogenic fungi isolated from different locality. So, a strain which will kill the insects fast, that means say a normal strain which is available currently is killing the insects 50% of the insects by 7 days. If there is a strain which kills the insect within 5 days, it is far better. So in those cases we measure LT50, lethal time, the time required to kill 50 percentage of the test organism at a specified dose or concentration. We can use either dose or concentration. Next comes knockdown dose, concentration or time. Here knockdown means here you might be which is the group of insecticides which cause knockdown toxicity. Pyrethroids, yes. Why only Abhishek and Rahul is answering? Sri Lakshmi, Shikha and Srisha, are you sleeping? No. What about Sri Lakshmi? Oh, yes. You are alive. Okay. Because I asked it, my daughter used to sleep in between online class. We used to wake her up during in between online class. If we are not taking care of her, he used to sleep in front of laptop. So that's why I asked. So knockdown concentration is main concentration dose or time is mainly used with the case of in the case of insecticides which have knockdown property. A typical example is pyrethroid. How much insecticides are knocked down? What is the concentration or dose of insecticide required to knock down 50% of the test animal? Next is effective dose or concentration. Effective dose or concentration EC50 or is ED50 is mainly used in the case of insect growth regulators, chemosterols, etc. Then coming to the what are the uses of bioassay? What are the applications of bioassay? Say we have a new pest. Say we have coconut spiraling whitefly, Aleurodicus rugoperculatus. It is recently introduced into our country, and we want to know which insecticide will be effective against this rugospiraling whitefly. Then we will conduct a lab study wherein we will screen large number of insecticides in the lab itself so as to come up with a list of potent insecticide. If you test 10 insecticides, of which 2 or 3 will be better, so that we can test only these 2 or 3 insecticide in the field. If we are directly going for a field study, we have to screen large number of insecticides in the field. It is time consuming, it takes up lot of cost. So in a short period we can evaluate the toxicity of the insecticide in the lab itself, and we can say that yes, this insecticide has better potential than other insecticides, so these insecticides can be tested in the field. So other pesticides which doesn't have much effect cannot be tested in the field. So that is screening of potential insecticides. Which insecticides are effective? So that is one thing. And the next application is fundamental studies on mode of action of insecticide. How the insecticides are acting, whether the insecticide is contact, whether they having contact toxicity, whether the insecticide is having stomach toxicity, whether the insecticide is having fumigate toxicity. This is studied by using bioassay. I will discuss about how we go about studying the contact toxicity or stomach toxicity or fumigant toxicity of a pesticide using bioassay when we discuss about various methods of bioassay. So the bioassay will help us to study the mode of action of insecticide. And next one is quality testing of formulations. Say if the formulation quinarphos 25 percentage EC, if the formulation is perfect, LD50 will be a particular quantity. If the formulation is substandard, it won't cause that particular mortality LC50 of the original or the a Paka formulation. So if the formulation is substandard, then we may have to go for higher concentration to get the 50% mortality. So it is used to test the quality of a pesticide formulation, that is active ingredient as well as adjuvant. So both we can test using bioassay. And the next step is generation of baseline data. Say I am introducing a new pesticide molecule, and before introducing the pesticide molecule into the market, I will check the mortality of a particular pesticide. I am releasing it against rice tempora. So I will carry out bioassay using stemborer larvae collected from different locations, and I will have a document on the LC50 or LD50 of that particular pesticide against tempora at that particular time before introduction of the time. This is just the ways to tell. You might have studied about certain. You might have seen certain advertisements for slimming purpose, to reduce the body weight before and after. That means before the person will be shown in extremely high body mass, and after using the drug, then the person will be extremely slim and all. In naptol and all, you might be seeing so many kind of things. So this generation of baseline data is something like that. That means before the introduction of the pesticide, what is the susceptibility of the insect to that particular insecticide? What is the LC50 or LD50? And after say 5 years of use of the pesticide, whether there is any change in the susceptibility of the insect to that particular insecticide. Say if what will happen if you are continuously using that pesticide for another 5 to 10 years, what will happen, whether the pest will be more susceptible or less susceptible? Less. It will be invariably less susceptible. So if you want to say that the pesticide is less susceptible, we should have a data beforehand. Unless and until we have a baseline data or a data before the introduction of the pesticide, we can't say that now there is a change in the susceptibility of a of the insect. Otherwise, we should have a susceptible population. Say now Anu Thomas is carrying out a work on resistance of brinjal fruit and shoot borer. against diamide insecticides. So we don't have a baseline data from Kerala. What is the LC50 of brinjal fruit and shoot borer to chlorantraniliprole or flubendiamide at the time of introduction of this two insecticides, flubendiamide and chlorantraniliprole. So what we are doing is we are taking a population from NBAR Bangalore where they are maintaining this population, this fruit and shoot borer population in the lab itself without any exposure to insecticide. So we can compare with the lab population, which is a susceptible population. So that is one thing that we normally do. Otherwise, it is better to have a baseline data. And next is detection of insecticide resistance. Say, you yourself are told that there will be a reduced susceptibility of the insecticide if you continuously use it. That means the insect has evolved resistance towards that insecticide. So bioassay can be used to determine whether the insect has evolved resistance to a particular insecticide. So the applications of bioassay include screening of pesticide, study on the mode of action of insecticides. We can check the quality of the formulation, we can generate baseline susceptibility data, we can detect insecticide resistance, and next we can determine the effect of synergist. Say I have found out a new molecule and I suspect that this molecule is having synergistic activity. So this can be checked using bioassay. This I will discuss what are synergists, how synergists change the susceptibility level and all when we discuss about synergists. Do you know? Does anyone know what is a synergist? Okay, no, yeah, then that's fine. Srisha, it increases the toxic activity. Yes, good. It increases the toxicity, but one thing is that synergist does not have it toxicity on its own. It enhances the activity, as Sri Lakshmi has pointed out, it enhances the toxicity of the insecticide, but that material, which is a synergist, does not have any activity at all. It doesn't cause any mortality in the insect. So that is a synergist. So that means if the insecticide, say quinalphos, quinalphos, when I use the quinalphos alone, the LC50 was 100 ppm. Then I found out a new chemical which I suspect that it is having a synergistic property. So I'll mix my discovery with quinalphos and I'll apply it to the insect. So earlier quinalphos alone we were getting 100 ppm as LC50, and if there is synergistic activity, the LC50 will be invariably lower than 100 ppm. Say I am getting 1 ppm of the as the LC50 if I am mixing with a synergist, then we can say that that chemical is having synergistic property. So in order to determine whether the chemical is having synergistic property, we go for bioassay. Then earlier we used to estimate insecticide residues using bioassay. That is called as microbioassay. A simple example that we that we normally see. I don't know who was practicing it. I used to hear from my batchmates who studied in College of Horticulture for their UG when we were doing the PG in IRA. My batchmate used to say that one of the professors from College of Horticulture used to purchase only those tomatoes which are infested with fruit borer. So he used to say that tomatoes are free from pesticide residues. So that is a kind of bioassay. That means we are leaving an insect to the vegetable. If the insect survives, yes, that it is free of pesticide residues. So that is another way of determining pesticide residues, but nowadays it is not at all practiced. But that is one application of pesticide insecticide bioassay. And then comparing the relative toxicity of the insecticide. Say we usually say that imidacloprid and thiamethoxam are effective against sucking pest. And yeah, one farmer has come to you, approached you, and he has said that I have a problem of cowpea aphid. please recommend an insecticide. So you know that for aphid management, imidacloprid and diamethoxam both are good. So which one should I recommend? Which is having more effectiveness or more toxicity to aphid? So what we will do is we will take aphid, cowpea aphid, conduct a bioassay with imidacloprid and conduct an LC50 with thiamethoxam. We'll compare the LC50. Say imidacloprid, the LC50 is 10 ppm and thiamethoxam, the LC50 is 5 ppm. Which one is more toxic? Imidacloprid has 10 ppm and thiamethoxam at 5 ppm. Which one is more toxic? Yes, it is thiamethoxam. So this is relative toxicity. We are comparing. Sometimes what we say that we used to have, you might have studied about check in the case of variety screening and all. In those your friends who are working in horticulture or agronomy, when they're carrying out a varietal screening and all, they will keep a local check. That means a known already recommended variety. Say if you are nowadays, whenever we are carrying out a varietal screening in rice in Kerala, we usually keep check either Jodi or Uma, which is the standard one. So in the same manner, related toxicity, if we are trying a new pesticide, we will keep the already recommended pesticide, already recommended widely used pesticide as this check or standard, and we will compare the LC50 with that. So that is carrying out studies on relative toxicity. Then we can determine the toxicity to natural enemies. That is what you have seen here. Pavitra Kumar is doing the toxicity of pesticide to Chilomenus maculatus. Then behavioral bioassay. We not only measure mortality, we also bioassay can be used to measure antioxidancy, growth regulator activity, repellent activity, etc. So that is behavioral bioassay. So in the case of botanicals and all, we may not get mortality. We may get only antioxidancy, growth regulator activity, insect growth regulator activity, repellency, etc. So those characters can be measured using bioassay. So bioassay has wide applications, wide use. So this is one of the typical feature with respect to pesticide toxicology. And what are the requirements of a bioassay? Before venturing into a bioassay, what should we do? The first and foremost thing is that we should standardize a method to carry out bioassay so that the results will be consistent and reproducible, because not all a single method is viable for all the insects. A single method is not viable for different kinds of insects. Say insecticides, I will say that aluminum phosphide, it is a fumigant. We cannot go for a bioassay of aluminum phosphide in a traditional way, as is the case of we carry out a bioassay for quinalphos, chlorpyrifos, or any pyrethroid. For aluminum phosphide, it is a fumigant. So we have to carry out bioassay wherein we can measure the response in such a way that the container is airtight, because the whatever the fumigant which is released, it should be in an airtight container. So we have to standardize a method so that it is suiting the insect as well as the insecticide, and it will produce a consistent and reproducible result. So different methods for different insects, different insecticides are standardized by WHO and Iraq. And another thing is that we have to standardize the biological conditions, such as that biological conditions means age of the insect, sex of the insect, size of the insect, weight of the insect, etc. We will discuss about it later in the last part. Then standardization of physical and chemical conditions such as temperature, humidity, photoperiod, etc. Here, one thing we can say that yesterday we have discussed about type 1 and type 2 pyrethroids. What is the difference between type 1 and type 2 pyrethroids? with respect to temperature. Do you remember yesterday's class? Type 1, no, Abhishek. Yeah, type 1 more toxic at low temperature. Yes, negative temperature coefficient. And so that means if we are carrying out a bioassay of a synthetic pyrethroid, type 1 or type 2 pyrethroid, within the 24-hour period, there will be a change in the temperature. During noon, there will be an increase in the temperature. At night, there will be decrease in the temperature. And if you are comparing type 1 and type 2 pyrethroid, there will be change in the result. So, whenever we are carrying out a bioassay, we should ensure that the temperature is maintained constant throughout the experimental period. So we usually maintain the bioassayed insects in a growth chamber, so that wherein the temperature is maintained the same, humidity is maintained the same, and photo period is also maintained the same, because toxicity can vary. The insect physiological activity can vary with different photo period. So whenever you look into any foreign papers, good quality papers with respect to bioassay, they will clearly say about the temperature at which the bioassay is carried out, humidity, and as well as photo period. So the standardization of age, sex, weight, etc. we'll discuss in the last slides. Coming to the different types of bioassay, I have discussed only major types of bioassay: topical method, injection method, dipping method, residual film method, fumigation method, and feeding method. Say, coming to the first one, topical application. This topical application, what we will do is, we will dissolve the insecticide in a non-toxic solvent. Non-toxic solvent means the solvent does not cause any harm to the insect. So that is one typical feature prerequisite for topical application. Insecticide is dissolved in a non-toxic volatile solvent. The example is acetone, and a particular quantity of insecticide is applied onto a specified location on the surface of the insect body. I will repeat. Say, so normally we used to apply 1 microliter, etc. That can vary with insects, but if we are fixing at 1 microliter of this pesticide dissolved in a volatile non-toxic solvent will be applied onto the insect body, and we will apply onto a specific part of the insect body. Normally, in the case of Spodoptera helicopa larva, we will apply onto the dorsal surface of the body. So that is one way of doing topical application. For carrying out this topical application, we have an instrument called as micro applicator. This micro applicator, this is the picture of a micro applicator. This is the place where we can fix the quantity of pesticide to be applied. We can fix 1, 2, 3, 4, 5, 6 microliter. That is in microliters. And here is a syringe. At the tip of this syringe, there is a needle. So if you are fixing the 1 microliter and pressing the button, the syringe will be pressed in such a manner that at the tip of the needle there will be exact 1 microliter of the pesticide. So that is the way which we normally carry out bioassay using a micro applicator. Uh, and usually we cannot, but is it LC50 or LD50? What is your opinion? If you are carrying out a topical bioassay, the result will be LC50 or LD50. There is a difference of opinion. Sri Lakshmi has told LD, Shika has told LC. What about the other three? LC, LC, Abhishek, LC. So only Sri Lakshmi has told LD. It's LC, it's not LD50 because we know the quantity of pesticide applied on the insect body, but we don't know what is the quantity of the pesticide entering into the insect body. So that is the major difference. So we know what is applied, but we don't know what is the quantity entered into the insect body. When we discuss about the next method, then you will be more familiar. And the result is expressed microliter or active ingredient per gram of the insect. If you are taking the insect body, you should no, sorry, this is the unit is different. It is in ppm. Topical application, it is percentage or ppm. We don't express it in body weight. So that is a wrong thing which I have given in the slide. When coming to the advantages and disadvantages of topical application, it produces highly precise, reproducible results because exact quantity of pesticide is delivered onto the insect. So the results are extremely precise and reproducible, and we can conduct large number of tests in a short time because it is pretty quick, fast, and the number of insects required is also low. This equipment is this micro applicator is comparatively cheap. A simple, inexpensive equipment. It doesn't require much maintenance and all. Hardly it costs about 1.5 to 2 lakhs. It's a simple one, inexpensive equipment. And the quantity of pesticide or the chemicals and solvents required is extremely low compared to other. That I will discuss whenever we discuss about other bioassay methods. Here, for each insect, we may hardly need 1 microliter. That is the quantity of pesticide needed. So the quantity of pesticide needed is extremely low. And the disadvantages include some species cannot be treated successfully. So if you want to go for bioassay using a top level application using a micro applicator, will you be able to go for bioassay with an aphid or a mite? Because you won't be able to handle it. You have to precisely apply that pesticide onto a particular part of the insect body. So very small, soft-bodied insects, we cannot use it. But don't think that it is not viable for small insects. I have seen the lab, that was the toxicology lab in the wherein I worked in IRA. That lab had a microapplicator, the same machine from the company called Burkhardt. This is the same machine. We routinely used to carry out topical application with different insecticides with almost all the storage pests, tribolium, bruchids, rhizoperta, cytophilus, etc. Because small insects can be handled. We usually don't handle with our fingers. We had a very small vacuum pump so that we can handle the insect without damaging it. So beetles and all we can handle it easily. This even if it is having smaller size. Then we cannot quantify. This is the answer for you, Sri Rashmi. We cannot quantify the exact amount of pesticide entering into the insect body. So this is with respect to topical application. Coming to the next method, that is injection method. In injection method, just like topical application, we will dissolve the pesticide in a non-toxic solvent, and this dissolved insecticide will be injected into the insect body. Usually this injection is carried out through abdominal sterna. That means ventral side of the abdomen. So whenever we are conducting a injection bioassay, we should be extremely careful that we should not cause any harm to the test insect. Because if you are causing any harm to the test insect, there will be mortality in the control also. So that we should take care of. So we should take care there should that there is no bleeding of the insect. And another thing that we should take care is that whatever the chemical, the solvent in which we dissolve the chemical should be non-toxic to the insect. If the solvent is toxic, means again it will affect the data. And whether what will be here, which will be kind of biocide is, whether it will give LC50 or LD50. Hmm? No answers. LD, 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 LD. Yes, it is LD50 because we know the exact quantity of pesticide entering into the insect body, and if we know the average body weight of the insect, we can say it as milligram per kg body weight. So that is one thing with injection method, and one few drawbacks with injection method is that it is very difficult to find a solvent which is non-toxic to the insect body. Even though we say that acetone is non-toxic, sometimes acetone may cause some toxicity to the insects. And the other thing, another thing is that size and behavior of the insects can affect the efficacy of this method. Take one example. You have to carry out a bioassay with rhinoceros beetle. Will you be able to go about with an injection method? You have to first design a needle which can pierce through the hardboard cuticle of the rhinoceros beetle. So the size, if it is extremely small, again injection will be difficult. So that is one thing which makes this method a little bit cumbersome. This time taking compared to topical application. Coming to dipping method, this is very simple. We'll prepare different concentration of the insecticide. We take the insect, we'll dip the insect in the insecticide solution for a predetermined time. That time should be constant for all the experiments, say 5 seconds, 10 seconds, etc. Keep the insect dipped in the insecticide solution for the prescribed time, and then remove it from the insecticide solution and keep it in clean containers and count the mortality. As the name indicates, that is dipping method. Then comes contact or residual exposure method. In contact or residual exposure method, what we'll do is we'll prepare different concentration of the insecticide. We will take a known volume of the insecticide and we will apply onto petri plate, and we will rotate the petri plate, and so that all the surface of the petri plate is having a uniform coating of the insecticide, and into that uh, uh, petri plate we will release the insect, so that the insect will move over the thin film of pesticide on the petri plate, and we will get the mortality data. This is the way to attain measure those insects. Say certain insecticides, if we apply onto the plant surface, and the insect may be hiding under the leaf surface, and once the spraying is over, insect may come to the upper surface and move over the treated surface, and while moving over the treated surface it pick up the toxicant. So this residual film method or contact exposure, residual exposure method or contact exposure method used to measure the toxicity of these kind of poisons. So here we will dissolve the insecticide in a solvent. We'll apply a thin film of pesticide on a petri plate, and we will allow it to evaporate, the solvent to evaporate, and we will leave the insects into the petri plate, and we'll calculate the mortality. In order to have a thin film of pesticide, we can use an instrument called a spottostover. There is a spottostover in our lab also, but it is an old model, it's a fabricated one, it's not a perfect one. Here what we'll do is, this is a platform wherein we keep the petri plate, and here through this we will apply the pesticide, and it is an extremely precise instrument, so that the pesticide, the petri plate which is kept on the platform will have a uniform coating of the pesticide. So that is one thing. So we will have a petri plate kept over here. It will give a uniform coating. We will remove the petri plate, allow the petri plate to dry, and then we will release the insect into it. This same potter stove can be used for topical application also. What we will do is, we will take a petri plate containing the insect. We will keep the petriplate containing insect in this platform. We will carry out the spray so that the pesticide will be uniformly sprayed over the insect body, and we will remove the insect and transfer it to a fresh petri plate. Otherwise, if you leave the insect in the petri plate itself, it will be a combined effect of residual toxicity and contact toxicity, because the petri plate is having pesticide residues. The insect will move around this petri plate and the toxicity will be enhanced. So, topical application can be also be carried out using Potter's tower. So, this is called as Potter's tower biosys. This is an instrument used to give same uniform pesticide spray on a surface or an insect. Coming to feeding method, here what we will do is I will take a typical example. So if we want to carry out a bioassay on Spodoptera litura, Spodoptera feeds very well on banana leaves or castor leaves, etc. What we will do is we will prepare different concentration of insecticide. We will take castor leaves or banana leaves. We will dip the leaves in the insecticide solution, leave it for air drying, and once the leaves are dried, we will keep it in a container or a petri plate and leave the insects into it. So once the insect feeds the treated leaves, it can lead to mortality, and we can measure the LC50 or LD50. So this is used to evaluate the toxicity of ingested chemicals, the pesticides which are taken in. So we have sprayed or dusted foliage or treated food or poison bait, etc. is provided. So Shahnas was working a feeding kind of biosay. What she did was she mixed different concentrations of pesticides in a food bait and provided that food bait to Spodoptera litura larvae. So that was extremely effective way of assessing the feeding toxicity of an insecticide. And before releasing the insect, we'll keep the insect starved for a few hours, because if the insects are allowed to feed till the bioassay, the insect, a few insects must have a stomach full, and a few insects must not have consumed food for say 2 to 3 hours. The feeding behavior of these two insects will be different. One insect which are not fed for 2 to 3 hours, once we are carrying out biopsy, starts feeding of the toxic pesticide uh, immediately, while one insect which has been completely fed may not consume the insecticide treated food for about next 2 to 3 hours. So the two insects, even though both are exposed to the same dose, may behave differently. So it is better to have a pre-starved insect for the bioassay. Otherwise, we cannot get uniformity. So this is with respect to feeding method. The next one is fumigation method. This is with respect to this is used for storage pests. The insects are released into a sealed airtight container, and the fumigant is released into it. Now I will ask one question. For assessing the toxicity of organophosphorus insecticide, which method will be more suited? You have studied about different bioassay method. For assessing the toxicity of an organophosphorus insecticide, which bioassay method is suited? There can be one or more, at least a few you can cite. No answers? We have discussed about topical application, injection, residual film method, dipping, feeding method, fumigant bioassay. Injection, okay, fine. Agreed, injection. All the method topical application, because it is a contact poison, we can go for topical application. Since it is most of the OPs are stomach poison, dipping can also be done. Feeding method can also be done. Then what about BT? You have a BT, Bacillus thuringiensis formulation. Which method? Feeding method. Abhishek said about feeding. Any other feeding? Feeding, feeding. Yes. Why we have to go for a feeding method in BT? Not we can go for dipping or topical application. It's a stomach poison. Yes, good. So now we understood how bioassays are used to determine the mode of action, how it is contact poison, how it is stomach poison, fumigant toxicity, etc. So this is the way bioassays are used. Depending upon the insecticide, we have to design a bioassay method. And coming to the sources of variability, though we may say that we may feel that bioassay is pretty simple, to get a pucker result, it is extremely difficult because even a slight variation can cause erroneous results. One source of variation is the age of the insect or the stage of the insect. If you want to carry out a bioessence Spodoptera litura, we have four stages: egg, larvae, pupae, and adult. Which one should we take? Larvae is one of the most susceptible stage. Eggs and pupae are less susceptible, so preferably we should go for the more susceptible larval stage. And another typical feature is it is the most damaging stage also. The other three stages are not damaging. And even in the larval stage, we have five instars. Which instar we should take? Because early larval instars are more susceptible to insecticides, while late larval instars are tolerant to insecticide. So we have to be carefully choose the age or stage of the insect for bioassay. Then coming to the sex, it's pretty difficult. Your senior Abinsha has carried out the bioassay with irradiation, gamma radiation, separating the males and females because the effect of radiation differs with the sex of the insect. In the same manner, insecticide toxicity can vary with sex of the insect also because female insects, because of the higher content of fat bodies, it is more tolerant to insecticides. So the sex is one reason, though we normally doesn't sex the insect and carry out separate biopsy. Only in specific instances we go about that. Then coming to the rearing temperature that we discussed, with changing temperature can affect the toxicity. We discussed about the example of synthetic pyrethroid. Coming to the food supply, that we discussed whether you for feeding biopsy we have to go for pre-starved insect. Another thing is that the quality and quantity of the diet in which the larvae is reared. A typical example I have given is that of a larvae fed with peppermint leaves and snap bean leaves. See, the any material which is ingested into the insect body, whether it is a pesticide or a plant metabolite, the insect body tries to metabolize it. Whatever, whether it is a plant derived chemical or an insecticide, the first reaction from an insect will be to detoxify that chemical, whether it is a plant derived plant metabolite or an insecticide. And the detoxification mechanisms for plant derived chemicals and insecticides are the same. Say, in peppermint leaves will have menthol, and we are treating the peppermint leaves with a pyrethroid insecticide and give it. So peppermint leaves or rear the larvae in peppermint leaves and then take the insect for bioassay. What will happen is that in order to detoxify the menthol content in the peppermint leaf, insect will, while rearing on it, the insect will release more detoxifying enzymes. And the same detoxifying enzymes are responsible for metabolizing the pesticides. So the enzymes responsible for metabolizing insecticides as well as plant secondary metabolites are one and the same. So this can alter the susceptibility. So the same larvae fed on peppermint leaves are more tolerant to insecticide than those reared on snapbean leaves because of the presence of secondary metabolites. Then coming to the next thing, that is population density. Say there was a discussion whenever we were discussing about the technical program of several students. We usually have a discussion about the number of insects to be kept in each replication. Usually, in tribolium and all, if you are going about bioassay in a small, in a usual petriblade, 9 centimeter petriblade, we usually keep 10 10 tribolium, 10 insects. We can go for 30 insects also. A petriplate can hold 30 tribolium adults also, but it will result to overcrowding and it can affect the toxicity. Another thing, if you go for Spodoptera litura, better in a petriplate limit the maximum number of insects to 4 to 5. If you increase more than that, it will lead to overcrowding and it will, the result will vary. And illumination, I told the photoperiod. Photoperiod can also alter the toxicity of the insecticide. So this is with respect to bioassay, applications, what are the factors affecting bioassay and all. Hope this is known, you understood what I have told you. Is it clear? Yes. So it's a pretty simple topic. There is nothing much to understand in that in this and all. So try to go through the materials I have given. Tomorrow I'll be taking probably, and most probably I'll take by 11 or 11:30. Okay, fine. Thank you.